Good afternoon, dear colleagues and participants of our meeting. My name is Bohdan Ostash, and I represent the team of Ivan Franko, National University of Lviv. And before we start to work on the seminar, let me briefly explain the purpose of our meeting. Our university is a participant of project Responsible Research and Innovation Ground in Practice in Biosciences, founded by European Union and Horizon 2020 program. And in the frame of the project, our university is a leader of the work package which deals with grounding actions on education. And it also is the participant of another work package named Ethics and Open Access. So several words about our project. The abbreviation of name of this project is RESBIOS, and RESBIOS aims to deeply embed in responsible research and innovation practice within 4 institutions in the field of biosciences from 4 European countries through the implementation of grounding actions of responsible research and innovations. And our university is one among these 4 institutions, and accordingly to strategy of RESBIOS, grounding actions are structured in 3 strands, respectively, aimed at first developing self-reflection on the changes needs in the organizations and dialoguing with potentially involved stakeholders for designing the experiments. Second aim is implementing the grounding actions in connection with societal actors in the territory and inside the research organizations. And last aim is developing permanent spaces for integration with society, implementing a set of institutional change inside the research organization, and reframing its governance settings. RESBIOS is focused on the biosciences sectors, which are one of the crossroads in the relations between sciences and society. And what about work package 3, led by our university? This work package is focused on the implementation of grounding actions related to education. These grounding actions aimed at broadening the educational outreach of implementing organizations, with a particular attention also to educational actors on the territory of intervention. That is why we invited Bohdan Ostash, professor of the Department of Genetics and Biotechnology, to participate in the seminar connected to work package 3, named Grounding Actions on Education. Dr. Bohdan Ostash graduated Ivan Franko National University of Lviv. He has experience of work in the abroad institutions such as Harvard Medical School, USA, at the Department of Microbiology and Molecular Genetics. Dr. Ostash was principal investigator of several domestic and international grants, and he is owner of 12 Ukrainian and 2 international patents. And the scientific interests of Dr. Ostash are bacterial genetics with an emphasis on class actinobacteria, chemistry and biochemistry of natural product biosynthesis, genetic and metabolic engineering, basic biochemical methods, and spectrometry. Also, Bohdan Ostash is a specialist in bioinformatics fields, and so now I invite Bohdan to present his short course in bioinformatics. Please, Bohdan, you can share your presentation, and we are ready to hear you.
Thanks, Elena. Do you hear me?
Yes, yes, we do.
Okay, thanks again. It's a pleasure for me to present a presentation on the topic already outlined by Elena. As already told by Elena, I am professor of genetics at the same university. So here is, if you'd like to know more about my topic or research interest, you can visit my ResearchGate page or our faculty or departmental page. At the main part is already taught. I am lecturer in bioinformatics. I lead a short summer course for masters, and that's why I think I am eligible to tell that I know a little bit of everything in the field of bioinformatics. Also, as a bacterial geneticist, I am familiar with genomes and genes and some basic bioinformatic tools. So here is my presentation, which probably summarizes both my teaching experience and also research experience. So the key part for me, and probably the biggest challenge for me, is both opportunities and perspectives. Let me tell you why. Bioinformatics is a kind of young discipline. I would say even the professor who gave first definition shown on the picture is alive, and she is still active professor at the University of Utrecht in Netherlands. So Pauline Hogek defined bioinformatics as follows. It's a very broad and concerned with certain molecular processes. It doesn't tell us what these processes are or what their molecular, I would say, foundations are. But still at the moment when this term was developed, you see, it's 1970, almost 50 years ago. This is pretty much what we could say about informatic processes. The availability of genome sequences was far away. 1977 is the first landmark paper on genome sequencing. Oh, sorry, not even genome, but just single gene sequencing. Now we know more, and now we can sort of come up with a more detailed definition of bioinformatics. And I personally like the one which I found on a web page of some NIH institute. I just highlighted the key words of this definition. So bioinformatics sort of deals with biological data, and these biological data, just let me know if everything goes wrong. I will try to fix it on my side. Okay, so here is the title page, and this is the second page. You can see this is a photo of Pauline Hogevec, who came up with the term bioinformatics and the first definition of what bioinformatics is. What I want to say is that our current understanding of bioinformatics is a little bit different. It's a bit more detailed and a bit more focused on analysis of biological data. And these data usually take shape of DNA or amino acid sequences. Of course, bioinformatics deals with more than DNA or amino acid data right now, but I would say this is the most traditional and the most developed topic of bioinformatics. So if we talk about DNA and protein, of course, we can't help mentioning the central dogma of molecular biology. I'm sure pretty much well understood everyone who is audience of this presentation. So the DNA is a carrier of genetic information going through the mRNA stage. It leads to proteins. And we can say the DNA is a genotype. So there's the amount of genetic information which can potentially lead to certain phenotype or morphological trait, which say the yellow spots on this lizard. So we go through the replication, transcription to translation. Of course, this is a necessary oversimplification. We know that not only DNA could be a carrier of genetic information in some viruses. RNA is a genome. Also, RNA itself could be a final product of genome. We know non-coding RNA, we know tRNA, and so on and so forth. But for the sake of simplicity and for the purpose of this presentation, I would say this central dogma properly embraces everything what we think the bioinformatics is. And it is also the paradigmatic model of the flow of information in biological system, the way that Pauline Hogeweg put it 50 years ago. So the information flows through from DNA through mRNA to protein. And there is no way for protein, there is no way for amino acid sequence to go back to DNA, although reverse transcription is possible. Now we know there are several molecular machines that mediate both transcription and translation. We will not touch this topic in much detail, but this is how this process looks like. So the bioinformatics deals with the text. So we go from a really cytological view of biology like this to more abstract bioinformatic view of biology. Each cell of human body, let's say more strictly, each somatic cell of human body carries a diploid set of chromosomes, and these chromosomes are actually a string of letters. For bioinformatician, it's just a sequence of letters. So, and this is the information that bioinformatician usually works with. Depicted on the slide is a tiny portion of human genome. And if you look at this text, you probably could share the same feeling as if you would look at the text written by unknown or foreign language. You just don't understand it unless you know the rules which are used to write the text. I mean, both grammar, vocabulary, syntax, and so on and so forth. Indeed, this string of 4 letters makes no sense unless you know some bits of molecular genetics. Once you know what stop codon is and start codon is, and the rules that govern the gene distribution and so on, and then slowly, one by one step, you will make sense of this sequence. But unless you know these rules, it's just nonsense. And bioinformatics is a discipline which primarily was developed because of demand to analyze that text. And this is a central part of my list, right? But let's then start from the top. We have the text and we have a number of problems with it. First of all, if you have a lot of genetic texts, be it just nucleotide sequences or amino acid sequences, you will need a certain way to store this information. It looks trivial if you have just a small piece of DNA, but if you have millions of genomes, and these genomes are quite sizable, you will need to come up with a unified solution how to sort of store this information. And often you need not only store it, you need to make sure that this information is secure. I mean, no one can steal it, no one can manipulate it or distort it. And also sometimes you need, when we are talking about humans and personal medicine, it's very important for you that this information remains anonymous. There is also one specialized problem which goes as a first line in my list. This is called a genome assembly. As I will tell you a few slides later, currently we don't sequence genome in its entirety. I mean, we cannot just sequence the entire chromosome from the letter 1 to the last letter. We first chop genome in short pieces, we sequence small pieces, and then we need to put together these pieces into genome. This is called genome assembly. And it's actually a huge bioinformatic problem to make this process errorless and fast. So when we have these genetic texts, we need to know how to generate them and how to store them. Then there is this thing of analysis. When you have the string of letters, you have to make sense of it. You need to recognize promoters, genes. You need to predict structures, both secondary and tertiary structures of the biomolecules encoded by this text. You want to know probably evolution of the genes encoded within these sequences, within this text, as I put it. You need to do so-called phylogenetic analysis. All these tasks, all these problems are bioinformatic ones, or they involve a heavy dose of bioinformatics. We need to know how to program. We need to come up with efficient algorithms, how to analyze, align, and so on and so forth. And there is a third problem, not a problem, but a task which bioinformaticians usually face, is a visualization of bioinformatic data. That might sound a little bit unbiological. I mean, not a problem for biologists, but in fact, when we are overwhelmed with a flood of data, we need to find a simple and intuitive way to convey this information in form of some figure. And it's not really simple. And without consultation with biologists, clinicians, patients, just common people who would be the end consumers of this information, it's not possible just for IT specialists, for computer scientists to provide a necessary level of expertise and service to visualize this data. So this, I would say, some kind of a cross-border problem. People from different disciplines should sit together and think hard about certain problems that stems from analysis of this seemingly simple string of 4 letters. And from this point, I will just give you a short overview of the most interesting topics of bioinformatics of current interest. I must admit that the choice of topic a little bit deliberate. It is dictated partly by my research interest, but I tried my best not to focus only on bacteria. I want to say that, of course, it's not possible to embrace all the diversity of problems that bioinformatics focuses on, but again, I will just reiterate, I tried to cherry pick those that probably are the most hot, or the hottest one at the moment. And let us begin with the genome assembly. As I already told you, if we have the chromosome, let's say human chromosome, like 80, 100 million base pairs or longer than that, there is no currently technique available on the market to sequence the entire chromosome from the first to the last letter without interruption. What we usually do, we fragment this chromosome. Actually, we don't work with a single molecule. We have a population of, we would say a clonal population, because the same identical copies of the same chromosome are present in the test tube. We break down the macromolecule into small pieces, and then we sequence these pieces from the end. We end up with short sequences like that. And then by looking at the overlapping regions, we try to reconstruct or assemble the initial molecule. What you should bear in your mind that the final result is a single continuous sequence of 4 letters. We always call it a model of genome. Why wouldn't we call it just a genome? Well, the problem is that this entire process is error prone. For sequencing, we use PCR. For assembly, we use some algorithms which are not perfect. And so our final assembly might carry some errors. it might not be 100% identical to the initial chromosome which existed within the cell. So we have a sort of our best effort to reproduce in computer the sequence of nucleotide, but we always should bear in the mind it's just probably the best approximation of what existed within the cell. And there is this constant effort in the research field to develop better methods, both in terms of the length of the sequences that we generate and in terms of quality of the data. Let's take a look at the most popular sequencing approaches right now. The Sanger sequencing was the first one. It is a technology developed over the course of 40 years, and it's very well developed, but it is designed to sequence short sequences like 1,000 base pairs, 1,200 base pairs, and the error rate is pretty impressive. We have just 0.1% error, which would mean out of 1,000 sequenced base pairs, only one probably could be error. Now, starting from 2005, there was a rise of next generation sequencing technologies. The Pyro sequencing 454 was the first one, then followed by Illumina sequencing and many other. And I would say that currently Illumina or reversible terminator sequencing is the most popular and the most mature technology on the market for high quality and high throughput sequencing. Actually, Illumina generates really short reads. So a single reaction which leads to 1,000 base pair fragment is called a read, right? So in Sanger sequencing, the length of read is around 1,000 base pairs, or as it is told in jargon, 1 KB, 1 kilo base pairs. For Illumina, we have 150 base pair read. You see they are shorter, and before development of these techniques, it was believed that short reads are bad. The shorter reads we have, the more difficult it is to assemble the genome. But actually, if you have a lot of short reads, it's even easier to assemble the genome. And you can see that we have both shorter reads and the higher error rate. These red lines, these red vertical lines denote the fraction of erroneous nucleotides in this sequence. Now, we also have technologies to generate really long reads. The Oxford nanopore sequencing gives you around 10,000, 30,000 base pairs, and the record for this technology is an impressive 2 million base pairs, but you have up to 70% errors. One would say, what is the use of such error-prone technique? Actually, sometimes you need really, really long reads to overcome the problem, which I'm going to introduce you on this slide. Human genome is full of repetitive sequences, and it is a big problem for human genomics, but it's even a worse, I would probably say, the worst problem for plant genomics. Some plants harbor up to 90% of their genome in the form of repetitive sequences. This is true for some pines, and it's really, really difficult to assemble such genomes. Let us see why. Let's say you have this kind of genome, and you can see here two repeats. These red blocks are identical, so they are repeated twice. Remember, we sequence our genome in some pieces. We can sequence this piece, or this piece, and this piece. And when we try to assemble them, we can stumble upon read one, which would have this structure. So we could sequence from this end and go up to the end of this first repeat. And we can sequence from this end and go up to the second repeat, but we never read this green zone. When we try to assemble them, it naturally for all bioinformatic tools to assemble these two reads like this, which is error, which is a mistake, because actually in Genome we have this kind of arrangement, but we would never recover the correct assembly unless we have a long read which would read through first through the first repeat and then through the second repeat. So this is the use of long sequencing techniques. Even if you have a lot of errors, you still have the information about the relative location of all these genomic parts within the genome. And a current approach towards the sequencing of complex genomes is a combination of high throughput short sequencing methods like Illumina and long read technologies like Oxford Nanopore and some other which I don't mention, but they, of course, these three technologies do not describe the entire field of sequencing. There are some other techniques. So by combining a huge volume of short reads and some amount of very long reads, one can assemble really challenging genomes like plant genomes. For someone who is interested in the area of genome assembly, I would probably recommend to visit this site. This is a very nice educational website on different bioinformatic topics, including both genome assembly. One could see what kind of algorithms are used to assemble genomes, what are the most interesting and unanswered or unaddressed problem. From my opinion, I looked at that website and I tried to solve some of these problems just out of curiosity. I would say that development of better algorithms for genome assembly is currently a very perspective field, because we will soon experience the flood of personal genomics. It becomes really cheap, really inexpensive to sequence one's genome. And once you have the genome, you will stumble upon the problem of correct assembly and then correct annotation. And I guess there will be a lot of development around this question, and any who has interest would probably feel like in the right place. I mean, you would devote your time to a really hot and relevant topic in the near future. So let's say we assembled the genome. Let's say we have these perfect algorithms, and actually you will end up with a huge amount of information. If we would like to print our genome, it would take the entire bookcase. Actually, this is a part of exhibition at the Wellcome Center in London. The Wellcome Center was one of the most active participants of the Human Genome Project, and they just printed the entire genome in these books. You can see a lot of text, but actually right now sequencing of a single human genome is not that expensive. It takes around $1,000 if it's done at some huge sequencing center. But the understanding of this genome, I mean, if you would like to see what all this information means, would take an expert analysis, and the cost of this expert analysis is around $20,000. After almost 20 years of research, this is what we know. And it's striking that only a small portion of it, around 2%, is devoted to protein coding genes. This was rather unexpected. Once the human genome was published around 2001, it was kind of surprise because people expected our genome to carry much more protein coding genes, but it is what it is. And actually it is not like an end of the story. Right now, there is a lot of development in the field of human genomics and human biology, and I will just briefly describe some recent developments, quite interesting. Let's say our genome is full of so-called genes for microproteins. Before, I would say 2015, small proteins less than 50 amino acid long were considered very rare in our genome. We do know about such proteins, some hormones and so on and so forth, but they would rather consider it an exception rather than the rule. But currently we know that our genome is full of short proteins which are involved in immune response, in the development of immune system, in neural activity and so forth. Actually, the discovery of this protein is a good illustration of interplay or cooperation between bioinformaticians and biochemists, or experimental biologists. Both needed each other. Biochemists suspected the presence of such short proteins, and bioinformaticians then developed efficient algorithms to look for such proteins genome-wide. Anyone interested in this hot topic of human biology, I direct them to this specific database developed for so-called devoted to short open reading frames, and also a recent review where the field of microproteins is described. Besides the microproteins, another interesting twist in biology of humans is the amount of mutations. One of the idea behind human genome sequencing was the development of very precise view of pathogenic mutations. The leaders of Human Genome Project expected to sort of build a database of all mutations that lead to heritable diseases. But it turned out that finding such mutations are a huge challenge, just because each new human genome carries more and more unknown mutations. If we find some variant, let's say there is some piece of genome and one can see either guanine or cytosine in that position, one would call this position a single nucleotide variant. This is different from SNP. It's a very popular term, SNP, but we can call such a change SNP only if we know the frequency of this change. So we need to have sort of a population data. If we don't know the frequency, it's more correct to call such a change just single nucleotide variants. Anyway, be it SNV or SNP, when we compare different populations, let's say we compare the population from China, Beijing, we compare population of white settlers from Utah or African population. With each new population, with each new genome, we find more and more new mutations. And currently we don't see the saturation of this process. It looks like from a population genetic point of view, human race went through some bottleneck, and then there was a fast rise of our population, and it leads to sort of explosion of new mutations which have no any relevance to the disease or health. Sort of silent, we would say, mutations. Not silent, but probably dispensable for certain processes. And then one needs to distinguish just a dispensable SNV from a pathogenic SNV. And that becomes a huge problem. Currently, researchers amassed all the information about these SNV and SNPs in a databases. One of the most comprehensive database is shown on the bottom of this slide. So how could we distinguish? So let's put it simply. We have a huge noise in our genomes. Let's say we could find 2 million SNPs which distinguish my genome from a genome of any other person. How could we find 1 or 2 mutations which would lead to some serious disease? How can we call this variant healthy, let's say, or sick variant? One would need really sophisticated statistical approaches, and one would need a huge scale of experimental work. Here I show you one example. This was a genome-wide association study devoted to finding the genetic causes of schizophrenia. Psychiatric diseases are the most difficult ones. It's not possible to model them on animals, and they are a huge economic burden for developed countries. And so Western governments pour a lot of money to understand and prevent schizophrenia. So researchers analyzed, you can see, a huge number of persons just to reveal 1 or 2 mutations which would be directly implicated in certain pathogenic process. And what you can see here is so-called Manhattan graph. So you on x-axis you see the chromosomes, and on a vertical axis you see the relevance of that mutations or significance of this mutation to the disease or pathogenic state that we study. Here it's a schizophrenia. And the higher the point is, the more significant it is. So one could say from this graph that somewhere in the chromosome 6, there is a mutation which is pervasive in samples taken from schizophrenia patients. But researchers found around 20,000 mutations associated to some degree with the schizophrenia, but only some of them that go through beyond this threshold are probably causal, those that cause schizophrenia. Of course, this kind of study was possible only due to really tight cooperation of over 3,000 clinicians, researchers, and bioinformaticians. And the amount of work behind this figure is really unimaginable. But again, it's very interesting. I would say this is also one of the very good opportunities for bioinformatics and bioinformaticians to develop an even better algorithm to improve them to the point that they would be used by every, I would say, diagnostic lab. Going from human genomics, let me shift the gears to bacteria. Besides the analysis of human genome, besides the problems associated with some heritable disease, we also have currently a very big challenge of antimicrobial resistance. I will not repeat what's shown on the slide. Suffice it to say that we will spend a lot of money in the future if we will not find the way to counter the threat of these escape bacteria. And how can we help as bioinformaticians to find better drugs or predict this antimicrobial resistance? From the diagnostic side, the current traditional approach to find or predict if the bacteria is resistant to drug is a microbiological one. You seed bacteria in liquid medium and you can find the minimal inhibitory concentration of certain drugs that kills this bug. But it takes several days and it's a long way. Genomics and bioinformatics offers a faster and probably more comprehensive look. So we could simply sequence a bacterium isolated from the patient, and using some artificial intelligence and bioinformatic approaches, we can sort of predict the levels of resistance of this bacterium to all marketed drugs. So what is, again, we again stumble upon this term, artificial intelligence. Simply put, is just a lot of linear algebra packed into the computer. And both artificial intelligence, machine learning, and deep learning are nested terms, which means they are just a subsection of some bigger field or bigger definition. Let's just demystify this term. But first of all, we need to know that this is also a very hot topic. It's even said that if there is no artificial intelligence in your plan or prospects, probably your discipline is not that hot or fashionable. This is also true about biology. Let us see how artificial intelligence can be used to predict antibiotic resistance of certain bacteria. Let's take this example. This is non-typhoidal salmonella. It's a very big threat in the United States, but also elsewhere in the world. Currently, the researchers whose publication I referred to had available almost 6,000 salmonella genome sequences, both antibiotic resistance and control samples. For each strain, they have metadata, which means it was known the source of this bacteria. Researchers knew the level of resistance to antibiotics for all of these bacteria. So these are metadata. And people tried to use these genomes for prediction, purely bioinformatic prediction of their resistance level. And how did they do that? They just cut the genome in small 10 nucleotide letters. So you can sort of divide your genomes into two sets, those coming from susceptible, wild-type genomes, and those that are already resistant to drugs. And simply, researchers looked for certain small 10 nucleotide words, which would be prevalent in resistant strains and absent in and absent in the control sample. Well, this is called a KMER approach. Okay, so actually it is impressive that their model, so they trained the computer, and their model was able to predict the level of resistance to all drugs of each strain with a 90% accuracy. Again, it's possible to see what KMER sequence was the most diagnostic one. Usually you can see really, let's say, trivial targets. One would expect that mutation in this gene would lead to some certain change in antibiotic resistance, but some of them were quite unexpected. And this again shows the power of artificial intelligence for finding a solution for a really important societal problem. Much like we use the bioinformatics to predict the resistance to antibiotics, we can use bioinformatics to find new drugs. Let's say we have some bacterium which produces, we sequence the genome, and then we can predict how many antibiotics this bacterium can produce. So bacteria can be a problem, but bacteria can also be an answer to this problem. Some bacteria are pathogenic, and now we're talking about bacteria that are able to produce antibiotics. Well, this field also is very interesting right now. Can we really predict all antibiotics that can be produced by certain bacterial genera? Let's say we sequence the genome of this bacterium, which is known for 20 years to produce only this drug, this molecule. But when we sequence the genome, we see around 40 groups of genes, also called gene clusters, involved in production of different molecules. And most of them are entirely unknown. And again, we need some tools which both identify the genes and visualize them, so that researcher can pick up the most interesting gene clusters. Actually, this is the field I am interested in, and where a few of my graduate students worked to build certain tools which would show how the gene clusters identified by different tools can be mapped to the single genome. I believe that this is also very interesting field. As long as we need to find new drugs, the development of new approaches and algorithms to find antibiotics would be in high demand. Of course, now we switch to another topic. The field of phylogenetic analysis is already very mature, but actually, although we have a lot of tools, they still are not comprehensive. They still don't give you a complete answer. Even for very simple trees like that, we would need a lot of time, actually unimaginable amount of time, to really check all possible variants, which means that current method of phylogenetic analysis are approximate. And as a recent global pandemic of coronavirus showed us, these data sets could be such large that all available bioinformatic and phylogenetic tools cannot really digest and visualize them. And I think we see the renaissance, we see the resurgence of interest into this very old field of phylogenetic analysis. It's like, again, because of big data. When you need to analyze just 20 sequences, we already have all tools to make it. But if you need to analyze 20,000 sequences, you probably need to build new tools. And this is where young researchers may come into play and apply their knowledge of artificial intelligence, IT technologies, and so on and so forth to really develop tools which completely explore the entire space of phylogenetic trees and not just randomly pick something which probably could be optimal, but might be just a local optimal in our search space. And the last one, and very fast, you probably know that even the simplest peptide can be encoded by a large number of synonymous sequences. Our genetic code is Degenerate, but we don't really know the rules, why certain genomes use this codon and another genome prefers another codon. There are many explanations for that. Some people say, well, probably some rare codons are slow. They allow ribosomes to move slowly at the beginning, and the translation initiation is the most important part of translation. But this is just one of the theories, and these theories are multiple. Actually, we don't have a good experimental evidence for that. But what I want to tell you is that synonymous mutations are very popular, are very distributed in our genomes, and we don't really know what they do. The default assumption is that synonymous mutations changes nothing. So synonymous mutation is sort of silent, but there are some good examples where synonymous mutation changes the behavior of the protein. So this is a protein involved in a resistance to certain anti-tumor drugs, and a single silent mutations makes this protein more efficient in a transfer of certain drugs and also more resistant to certain inhibitors of this protein. There is a good biophysical explanation of this, how synonymous mutation may change the dynamics of protein folding. And I will not go into this problem because I already took a lot of time. I just want to tell you that we have several very interesting and intriguing questions in biology where the bioinformatics would be of really great help, because bioinformatics can take huge data sets, can model them, and can come up with some interesting answers for the questions that are shown on the bottom of the slide. And now let me end with a question which could be not question, but an assumption or statement, which is bittersweet depending where you are. If you are more biologist, you would say the biology becomes digital science is not good. But if you're a bioinformatician, you would say, well, it's good. It's actually what I would expect in my career. But I guess the truth is in the middle. We need to talk to each other and understand that neither biologist, I mean pure biologist, experimental biologist, nor bioinformatician could not exist in isolation. We need to talk to each other, and it just will be for better of the science as a whole. Thank you for your attention and patience. I will be happy to take your questions.